Our next speaker is Maurizio Rossetto. I've known Maurizio for a long time, since he was in WA many years ago. Maurizio heads the Research Centre for Ecosystem Resilience at the Botanic Gardens of Sydney. The centre team integrates evolutionary, ecological and environmental knowledge to understand why plants are distributed and assembled the way they are and to guide applied flora management and conservation outcomes. Today Maurizio is going to talk on conservation genetics, an indispensable tool providing multiple practical solutions across multiple scenarios. Thanks, Maurizio.
Thank you. Thank you. Yes, it has been a very long time since I was in WA, but great times. Before I start, I want to thank Adrian this morning for welcoming us to country and also acknowledge all the traditional custodians of all the land that we live and work on. As David just mentioned, one of the things that the Research Centre for Ecosystem Resilience does is to provide genetic information in support of applied threatened species management outcomes. And so what I want to do is remind you all why it is critical to have that information when we want to have applied management outcomes. So maybe as a starting point, if there has to be a take-home message, is that we have to remind ourselves that conservation is a long-term game. So we really need to focus on quality rather than quantity. It's not about the fact that I've planted 1,000 trees out there or that I've propagated 200 individuals in the nurseries. It's about ensuring that those 2,000 trees and those 200 propagules will lead to self-sustaining populations. And this came across in many of the discussions that we had today. And by self-sustaining population, we mean populations that are able to reproduce and be resilient now and in the future, well beyond our capacity to monitor, at least our personal capacity to monitor them. And to have this capacity to do that, to have that qualitative measure, we need to have some sort of genetic information. And so this leads me to the second point, that we should really start our measurement strategy, our applied management strategy, with the genetic information. Not leave gathering genetic data as an afterthought. I've got a bit of money left over at the end of the financial year. Why don't we do some genetics? What genetics? What exactly are your questions? And this leads me to the third main point, that we need to stop saying that we don't have time and we don't have money to do genetic work. It's nonsense. We have plenty of time. There is nothing stopping us from continuing with our on-ground activity while we gather information, whatever that information might be. And if we gather that information from the beginning of the project, if we have done something wrong in the in our preliminary applied management strategies, we can correct it. We can correct it fast, we can correct it properly, and we don't have 10 years of remediation to do. So we have time and we have money. Oops, let me just change to the next one. This is just coming out a figure from a paper that was just accepted work that Chantal Doyle did, where we looked at the amount of money that we spent, that was given to us to do genetic study, and what was the proportion of the money in relation to the overall expenditure for that particular project. And as you can see, the amount is actually quite small. And it's most likely that it's actually an overestimate, because the reality is the proportion is about half of that, because those additional costs are just the costs that are publicly available, and that's just the expenditure that the Saving Our Species program has done for those particular species. So there's a lot of in-count, there's no count, there's a lot of contribution by community groups, there's a lot of contribution by academia that are counted there. So the reality is that genetic value is probably even smaller than that. So the money is there. It's not a question of the money is not there. And if you start with the idea that you need to have that information as part of the process, then you make it as your starting point. The other important thing to consider is that genetics, genomics have really changed. So the technology is really advanced so that we can get a lot of information much, much faster and much, much more cost effectively than we used to be able to do. And we can do that if we think about that with the economy of scale. So we don't think about one species at a time, but we think about it in the way where we looked at many, many species at the same time. And if we follow specific workflows that are planned to give you the answers for the specific practical questions that you want, you don't have to learn all there is to learn about the species. You need to have information, the information that you need. You can plan a single sampling strategy and a single round of analysis that will provide you all those answers. It will provide you answers about taxonomy, about diversity, about hybridization, about ploidy. What species, what individual should you use for your translocation? Is there hybridization happening? And so on, all in the one go. So really the opportunities are there. Let me just show a few examples to just illustrate the type of issues and how do we resolve those issues and how we could, if we would have had that information from the start, we would have avoided some of the errors that have happened through time. So the first example I want to use is one that has already been mentioned a little bit and is what we call taxonomic inflation. Unfortunately, and I must say we were a bit surprised when we started to do this work on not how common, but on the number of times where species were described and listed before any proper genetic work was done to determine and validate the fact they were a species. Now think about it. If you name something that is not a species, you have two issues that you have to deal with. First of all, you might be not wasting, but you are spending time and money that is already limited, right? We have already so many threatened species, and you're adding another species and another species. But also you are, by constraining that group of population or that population to what it is, that lineage to what it is, you're actually compounding the problems rather than resolving the problems. Anyway, a typical example is Banksea vincentia. Banksea vincentia was one of the most probably threatened species in New South Wales, if not in Australia. It was just a dozen or so individuals in Vincentia. And although a lot of really good work was done to try to manage the species and to propagate and to look after it through the years, there was never an absolute certainty. There was always a little bit of lingering doubt if it was a species or not. And so eventually we were asked to do a genetic study. And so we put Banksea vincentia and we analyzed it across with all the related species. And to cut a long story short, what we found, is this pointer going? Oops, no. Nope. Panic. So what we found is that when we looked at that genetic data, is that Banksea vincentia was nested within Banksea neoanglica. So Banksea vincentia is not a species. But not only that, what the genetic data showed is that it's not even a disjunct population that could have some value for conservation. We could actually see that it's likely to have been planted from a population in Bunu Bunu. So in other words, a lot of time, effort and money and goodwill was spent to look after a plant that was probably planted not long ago in Vincentia. And incidentally, there was also really quite a lot of ex situ collection that had been made and a lot of seeds that have been conserved, and all the seeds that were germinated were shown to be hybrids. So really, it was a lot of goodwill and a lot of money that was spent, unfortunately, for the wrong reasons. Now, speaking of ex situ collections, another species from the general area of southern New South Wales, can't even talk now, is Daphnandra johnsonia. So Daphnandra johnsonia is a rainforest tree, and it's restricted to multiple tiny fragmented populations. And because of that, again, a lot of goodwill, and I should add that a lot of those populations are sterile, they're not producing seed. So again, a lot of goodwill, a lot of effort, a lot of research went behind trying to understand why that was the case. Ex situ collection were developed. But there was, again, always the doubt, not that it wasn't a species, it was a species, and we did test that, and it was a species. But there was a suggestion that there was clonality, but it wasn't really visible because some of the stem can be quite separated, quite apart. Anyway, again, to cut a long story short, eventually they asked us to do a genetic study many years after all those other processes that had gone on. And what we found, what the graph is showing, is that wherever you see a red square, it means that the population is clonal. So most of the population were single clones. So two consequences of that. Number one, that's the reason why they're not fertile. So a lot of studies on pollination, the availability of pollinators, on diseases, on all sorts of other issues, but turns out that the only populations that were actually producing viable seeds were the ones that had high genetic diversity. All the ones that were clonal were not producing any seed, which is a good thing, right? Because now it gives us a good, clear path ahead how to translocate genetic rescue, and we can reestablish a living population. The other issue was a lot of the material that had been propagated, and a lot of time and an effort in spending propagating this material, was actually clonal. So some of the collections were single clones. So a lot of time and effort had been spent in propagating a single clone rather than achieving the purpose, which was to preserve genetic material for the long term. But now we've got the information, and we can revert all of that and deal with that. So just quickly, another couple of examples. So speaking of translocation, so if we want to do a translocation and a guiding and trying to support genetic rescue, then we can actually identify what are the best individuals to do that. Because it's not just a question, if you have a population with low diversity, say 20 or 30 individuals, it's not just a question of taking those 20 individuals and plonking them somewhere else. You're just going to recreate exactly the same inbreeding issues. You have to identify what are the most diverse individuals. You have to minimize related and kinship, and then you can kickstart the process again. So a quick example here is Procentera densa. Again, another one, there was a taxonomic issue, but it was all legit, so that's great to know. And what we found is that we had two slightly genetically differentiated groups of population, the northern ones in red there and the blue ones. And then we could identify the number, the individuals that would maximize diversity and reduce kinship and provide scenarios. We could provide a translocation scenario for the red group only, for the blue group only, or for a mixture. Again, the other important thing to remember there is that by using that information, we can really minimize the amount of effort, because the number of individuals that you need to propagate to achieve that is much smaller than what you would think. So again, it's about maximizing the outcome from the limited resources. And then once you've done your genetic rescue, of course, you want to check that you're actually achieving what you're planning to do. And so you can monitor, not just monitor that they're producing seed, but monitor what it is that you are getting out of those seeds. And this is work that we did some genetic work on this particular species, Fontaneraria, which is a little rainforest species from northern New South Wales, 20 or so years ago. And since then, Di Brown, which is here amongst us, has been re-establishing translocated plots where she was maximizing genetic diversity based on the genetic data. And now we're in the position where the first lots of seedling, in fact, it's quite prolific from what I understand, it's very prolific, it's going wild, are being produced, and we can check those seeds through. And what we are finding is not only we're getting all the different combinations from all the different individuals we were not getting before, but we can also see that some individuals are contributing more than others, which is what you would expect through the years. And maybe that information does not only tell us what's going on, but it can help us manage those seedlings. So if we want to move the seedlings somewhere else to do new population, we know that we can use some but not others, so that we don't recreate all those inbreeding problems. So hopefully, and although I've been running through this, but hopefully you can see that there are lots and lots of applied outcomes, and you can see why it is critical to really start your threatened species management processes with that information, because it'll make things better, it'll make you go faster, and will save you money in the long term. And the other thing, the last point that I want to close in, we can do this at scale, right? You know, people used to tell me, you can't do this for every single species. Yes, we can, and we are doing that. So we've already, in the last 5 years, we've already done about 20% of the flora. So we've started working on more than 140 species. We already produced 44 reports, applied reports that cover 60 plus species, and more than 20 of them have resulted already in applied outcomes, and so on. This is not just, look at us, it's just, this can be done and should be done. And although here I'm talking about, obviously we're dealing mostly with the New South Wales flora, the same thing now can happen in Queensland as well. Queensland has just employed a conservation geneticist, Patrick, that will talk, will give a presentation later. So this system is replicable everywhere. And I'll leave you with this. Thank you.
Thanks very much, Maurizio. It was great.